Okay, in this segment, I just want to talk about how population genetics and diversity impacts pharmacogenomics in general. And it all derives from some of the conversations we've had previously on the basics of genomics. First one to say is that we must make sure that SNP or variant associations with drug response phenotypes, we have to be very clear whether they are simply associations that are statistical, or are they the causal variants. We've talked about this a little previously. In general, when we look at pharmacogenes, it's the first time I've used that phrase in a while. A pharmacogene is a gene in the genome that is involved in either the pharmacodynamic or pharmacokinetic pathways for a drug. When we look at a pharmacogene, there are ways to tell, there are hints for whether it's likely to be associated, just an association or causal. For example, if the variant changes an amino acid, and the change is what we would call not conservative. It changes the amino acid to an amino acid that is very different. So for example, the amino acid D is negatively charged. If you change it to the amino acid W, which is tryptophan, it's very hydrophobic. This would be a big deal. This is much bigger than this, and it's uncharged versus this. So if it's not conservative, and if it's in the amino acid coding region, that would increase the chance that it's causal, but you still need to do experiments to prove that. If it doesn't change the amino acid, it could still be the causative variant. So you could imagine a variant that changes the stability of the messenger RNA. Stability of messenger RNA. So it may or may not be amino acid changing. It may be actually synonymous. That is to say, it may not change any amino acids, but the introduction of that new base in the mRNA leads to more rapid degradation, and therefore the availability of that gene to do its job changes dramatically. The other possibility is that the variant is not in the protein coding region. This, by the way, could be in coding or non-coding. Another possibility is that the variant affects the transcription of the gene. So it might be in a so-called promoter or enhancer, which are non-coding DNA elements that attract the transcriptional machinery to make the gene. So it's often drawn like this. Here's our gene. Here we have DNA promoters and enhancers. A transcription factor, we'll call it TF for transcription factor, sits, recognizes a certain DNA sequence, and recruits the polymerase, which I'll just call Pol, to start making an mRNA transcript. If there are mutations, or polymorphisms is really the right word to use, if there are polymorphisms in the TF binding section of DNA, it may bind more or less tightly, and that may change its ability to influence the polymerase to start transcribing. So these are the typical causal stories that make an association into a story. But none of this can typically be done purely computationally. This normally has to be done with experiments. Second big idea from population genetics, and of course, the reason they may be associated is because of LD. So LD leads to the statistical associations, but I would say real biology leads to the causal variants. The other important thing is the ability to be, you don't want to be tricked by ethnicity. So that's a funny phrase. What do I mean by that? We know because different populations have different SNPs. So here, populations have different SNP frequencies. Populations, they all share most of the SNPs, but their frequencies can be different. So populations have different SNP allele frequencies. That's fine. But guess what? Populations also have differences in their disease risk. So populations have different disease risk. And guess what? They also have different drug response phenotypes. Now here's the problem. Drug response phenotypes. It is easy to do a study where you find an association between a SNP and, let's say, a drug response phenotype, a drug phenotype, that is totally wrong and artifactual and is a product of the fact that because this population has that phenotype more commonly, and because it also has this SNP more commonly, you have a false association. And it really is a statistical mistake in your study design, statistical error, which makes you say, oh, look, whenever I have this allele, I seem to have this phenotype. I have an enrichment of this allele for this phenotype, where actually what's happening is this population has, for two separate reasons, it has this allele more frequently, it also has this phenotype more frequently, and you're detecting that correlation, which has absolutely nothing to do with the underlying biology. It's not even giving you the right segment of the genome. It's purely giving you a spurious correlation. That's the word I was looking for, spurious. Therefore, when you are doing any kind of genetic study, you must be very careful to match the ethnicity of the cases and the controls, the discovery group, so that you don't discover things based on spurious statistics. And there are many examples in the literature of this happening. And it leads to the third point, which is a very vexing problem, which is that the world is cosmopolitan. That's a big idea. I will explain why I'm talking about this. About 11,000 years ago, most of the initial human migrations ended. Initial migrations ended. And then we could have defined all of the different populations that we might want to study. You could define Europeans, Southeast Asians, Africans, South Americans, fine. But guess what? In the last 11,000 years, we haven't stopped. And in fact, airplanes and boats now allow all these populations to mix. So that currently, most people do not have ancestry entirely derived from one of those initial migration groups. And it leads to this idea of admixture. So I'm from New York. New York is a melting pot. I have ancestors from, well, certainly all over Europe and even beyond. If you go to San Francisco, New York, people have flown in, boated in, walked in, bicycled in. And so their chromosomes are highly admixed. And so what we're seeing is that a lot of the associations that we find need to be checked in the context of populations where the LD structure might be different. So the LD structure is different. Kind of a corollary to that is the haplotypes that are present are different. And therefore, any pharmacogenomic reasoning based on association is going to be very dangerous, and you really need to focus on causative variants, because at the end of the day, you'll have to check these genomes for the causative variants. You will not be able to rely on the correlations that might have been present 11,000 years ago, but have all been messed up in a good way, mixed together, and destroyed. So causal variants become key. And any kind of associations that are statistical become increasingly unreliable. This is made worse because current pharmacogenomic knowledge, in one study that I've seen, more than 90% of pharmacogenomics papers are on European populations. That's great for the Europeans, but it means that for all other populations, and for people who are European but have a mixed ancestry, the associations are likely not to hold up, whereas the causal variations are likely to be money. So there's a huge emphasis on finding the cause of these phenotypes and not simply being satisfied with the associated SNPs. Very important point. So the point to write here is that because 90% of pharmacogenomics is done in European populations, they have unclear applicability to both other populations, including admixed populations.